Hello, I'm Claire Prentice, Benchling Lead Analyst at DigitalRadius, and today I'll be talking about who we are here at DigitalRadius and sharing information and best practices for our favorite cloud-based ELN and LIMS platform, Benchling. Here at DigitalRadius, we take pride in being your digital transformation partner. We help companies of all sizes across all industries transition from more archaic paper or Excel-based processes and workflows to new, modern digital technologies to improve their efficiency, competitiveness, adaptability, and move faster as a business. Our founder, Ander Talat, was the former VP of Enterprise Systems at Moderna and then Resilience, and all members of our R&D IT team come from a research background. We have a deep understanding of the R&D industry and can empathize with the challenges of scaling and bringing drugs to market, having experienced those challenges ourselves. Today I'll speak on some current challenges in R&D that impact data management and require a digital solution, review the Benchling applications listed below, and wrap up with Benchling best practices, including a demonstration of workflows. Let's jump into some industry trends that require a modern solution. We'll start with the complexity of science. Currently, legacy software is not built for modern science or the needs of scientists. Scientists rely on various tools, platforms, softwares, and databases to perform and design their experiments, molecules, and drugs. Companies and researchers are generating an enormous amount of scientific data, but they lack the ability to integrate that data and information at scale, apply analytics, or AI or machine learning. With the increasing specialization within and across teams in an organization, there comes the challenge of merging workflows or organizing handoffs. Workflows require significant collaboration and integration into a fragmented or legacy software ecosystem. Especially in the case of acquisitions, this proves to be a challenge. You may recognize some of these challenges in your own organization, and that is why I want to talk today about Benchling. Benchling is a cloud-based ELN and LIMS platform that provides solutions to these challenges. So let's introduce you to Benchling. This cloud-based ELN and LIMS platform designed by scientists for scientists has quickly become the gold standard for ELNs within R&D for a reason. Benchling is intentional about creating a platform focused on digital transformation, allowing companies to stay on top of their data and scale. Benchling is moving us away from antiquated physical lab notebooks, Excel, and email to their integrated platform. This graphic illustrates Benchling's various application and important platform characteristics. What they describe as their productivity hub includes 7 applications and lab automation capacity. The left side of this arch includes their foundational applications that are included in most packages: the notebook, registry, inventory, and biomolecule design. These 4 applications alone provide your team with the tools to design molecules, DNA, RNA, and protein, track samples through the utilization of registry and inventory applications, and capture structured and unstructured lab work using the notebook application. Moving to the right side of this arch are more recent applications, including workflows, studies, insights, or analytics, and then also their lab automation. Not only can you capture data in Benchling, but you can build your processes into Benchling, allowing for smoother communication and handoff between teams and capturing all data in one place that is easily searchable and accessible. Now that we have a general understanding of the Benchling platform, I'm going to dive into each of the applications to talk about their specific capabilities and functionalities. We'll start with the notebook application with a focus on templates. The notebook is foundational in Benchling and captures both structured and unstructured data in the form of entries. You can view this application as you would your physical lab notebook. For routine protocols, admins can create templates like the one shown here. Creating templates allows for easy management of permissions and access. Administrators are the only ones who can create templates, and they can also determine who and how other users interact with that template. This aids in protocol version control. Notebook entries can also be submitted for review and sign-off to replicate the witnessing aspects of the physical lab notebook. Additionally, users can configure media and buffer prep calculation tables into templates to ensure standardized buffer prep. A newer feature includes links to procurement sites to ensure you have the correct reagent and know where to order more when you're almost out. Let's talk about the molecular biology suite. With this application, you can visualize DNA, RNA, and amino acids. You can upload your own sequences or upload from a database. So for example, we uploaded this plasmid sequence here. And once uploaded, you can perform sequence analysis, do sequence alignments, create oligos, optimize cloning, annotate your plasmids, run a digest, and more. Through Benchling, you can standardize sequence registration by including required fields of information such as antibiotic resistance, backbone source, and more, whatever you need to know. Again, like many other of the Benchling applications, this app is searchable with version and permission control. All right, let's talk about the registry. While conceptually straightforward, the registry is critical for standardizing and tracking data within Benchling. The registry consists of entities which are customizable to fit the type of data and samples you would like to capture within Benchling. Examples of entities include plasmids, antibodies, cell lines, and mixtures, but the possibilities are truly endless. Once you register an entity, you can create material genealogy through parent-child relationships. This is an important feature if you would like to keep track of sample lineages or if you have multiple aliquots and need to enforce those lineages. Once registered, all entities are searchable and queryable. The inventory application creates a digital representation of your physical storage systems. Typically, labs choose to recreate their freezers or dewars within Benchling. However, you can create any storage or locations that make sense for your physical space. For example, within this inventory, we've created a building 1, lab A and B. If you want to create more custom locations, you would go to location schemas and create schemas for those particular locations or storage systems. What makes this application powerful is its capability for material management. This is critically important if you have multiple lab users or groups that are sharing material. This application also dovetails nicely with the registry. So once an entity is registered, it can be given a place within the inventory. Additional information such as concentration and volume can be added so users know how much and where a sample is. This limits the number of headaches and emails between colleagues asking where the sample is and why there's only a microliter left. If your lab has a particular nomenclature, you can carry that into Benchling, or you can utilize the labeling and barcoding features within this application. The Workflows application streamlines your processes and handoffs between teams. Through this application, you can create a workflow for a particular process. This can be anything from protein production and purification to QC testing. So here on the right, we have an example workflow for a QC test. Each task becomes part of the flowchart and can be assigned to specific users. Once completed with a task, the next user assigned to the next task is notified and proceeds with the sample. If your team is already using the registry and inventory, that information can be added to workflows so everyone with hands on the process knows where the sample is and how much is left. This allows users to determine what stage of the process a sample is at and lends to smoother handoffs. Workflows can also be used as a request system, whether that request is for solution for material preparation or analytical testing. The request includes an approval management system. Later on, we'll explore an example of a material request workflow in detail. Insights is your application for all things analytical. You can easily create dashboards and reports to see how your end users are using Benchling to review bottlenecks or gaps in your processes. Insights is a valuable tool for reporting and data visualization, so you can pull data across multiple entries to compare lots of an antibody's binding curve, for example, or track cell viability over time or passage number. For large data sets or analytics, you can run SQL query to retrieve wanted data from insights, although you can always download notebook entries without any coding. Benchling's data warehouse is another option for data storage, and you can view structured data through SQL queries as well. The last application is Studies. This is Benchling's newest release and designed with mouse studies in mind. Through this application, you will set study standards and nomenclature, create task templates, and determine how you want to structure and collect data throughout your study. This application is valuable if you have a vivarium, as you can link barcoding and cage cards for easy identification of samples in mice in Benchling. All right, now I'm going to move on to some best practices in Benchling. I've broken our best practices down into 3 main categories. The first is to standardize early. So within your organization, you want to think about how you want to organize your data, whether that be through study type, your programs, team, scale, etc. Whatever you decide, this will become your data model and your guide for data organization in Benchling. When it comes to standardizing, a good place to start is within the notebook application. So you want to create notebook templates for routine assays and experiments, and create a standard way of implementing notebook protocols and nomenclature. Additionally, create workflows to automate and standardize work processes and handoffs. Our second theme here is to determine permissions and access. To start, you want to create a group of people that have global permission controls and access. These are your administrators. From there, the administrators can determine who else can access or create certain types of data and what is visible to the end user. Our last main theme here is to commit to upkeep. Schedule a time every 6 to 12 months to archive or retire any unused templates or entries. Create registration tables into your workflows for seamless entity registration. And finally, create data models and schemas in the registry with entity links. Through entity links, you can see all of the results linked to that particular entity and samples that are also associated to that entity. So I wanted to give an example of how you can standardize your data model, and that is through determining your project folder structure. This is something done during implementation, so if it was missed, I would start here. The project is the highest level of organization in Benchling. Project folders help keep your data organized, and there are specific permissions on who can create projects and folders. These are usually left to the administrators. So here are 4 examples of project folder structures. You have the combination, target focused, process focused, and team focused. Within each one of these examples, the project is the higher level of organization, and then you have folders and subfolders within that project. So we can move through these examples. So for a combination, you would have a name of the disease, target, team, assay, etc. Then within the folder, you could organize data within different folders as data, analysis, clinical site. And from there, say for the clinical site, we have multiple clinical sites. Moving on to the target-focused project structure, your projects are organized by a specific target. So an example would be HER2 or GLUT1. Your folders from there would be organized by team. So you have your therapeutic discovering team or immunology. Your subfolder from each team would be various molecule targets that you're working on. So molecule target 1, 2, etc. A process-focused structure, the project is the company function. So you would have assay development or therapeutic discovery. The folder from there are different components, so clinical data analysis, and the subfolder from there is perhaps different assays in terms of like a data analysis. Finally, we have a team-focused structure. So each project will be a different team within the organization. For example, molecular team, in vivo, clinical. Then the folder will be the target, for example, HER2 or GLUT1, and the subfolder will vary. So perhaps your target, within the target folders, you have your assays, clinical data analysis, etc. Before we jump into our workflows demo, I want to give a list of best practices for workflows. Our first is to include steps for rejected or expired tasks. This will help keep your workflows current and up to date. Second, assign a group of approvers for approval to reduce the amount of bottlenecks in the approval process. That way you're not just waiting on one person to approve all of the requests. Next, create task groups to easily swap and update workflows. This could be people that are familiar with the workflow process or team leads, likely people that have administrator privileges. Next, set a time to reevaluate processes and workflows every 6 to 12 months. Again, this will just help keep your workflows current, up to date, and streamline when necessary. Lastly, you want to set appropriate email notification triggers. Since all tasks are sent out via email, you want to create a distribution list for those email recipients to make sure that everything is timely and going to the correct end user. All right, we've made it to the workflow demo. So with workflows, you can standardize request fulfillment processes, which we'll demo today. You can manage assay requests and connect processes in a research pipeline. So the example we are going to work through is a request fulfillment process, and we're going to start from the perspective of the requester. So let's imagine you are on the characterization team and need to request drug material for a study from the production team. So you go into Benchling and request the material or materials through the following steps. So number one, you're going to go to this plus here and down to the workflow tasks and create a workflow task. So in this case, we want a material request from an internal source. There, so we're going to click on that. So after we click on the request, it will create a task group. So it'll open up this window, as you can see here, and what you're going to fill in is this required information. So your material, the date you need it by, amount, and your project. So this is what your request will look like once it's all filled out. It will create this task column here. It'll have a status, so active, and then this is all the information that you had previously added. So the material, date needed, amount, and project. And then you can, this gets sent to the approver. And that's it. So it'll pop up this flowchart here. You'll have these 5 tasks that need to be done, and now you will wait for the approval from the other team. So once the request is made, it must be approved. So we're going to switch perspectives here. So let's say that I'm on the production team and responsible for going through the material requests. So what I'm going to do is come up to the search bar here and search for all material with a pending status to see what needs to be approved. So that is what I did here through applying those filters. And now I look to this column that says assignee, and I see that these are currently unassigned. So what I'm going to do is assign those materials to a person within the production team. All right, so I'm going to first open a request, and then I will assign the task. So this is a searchable field here, and I will pick this end user to perform the task. And then all I do is click execute for the task. Then the assignee, so this user here, will receive an email like this one informing them that they have a task request. All right, so I was assigned the material. Once the material is produced and ready to hand off back to the requester, the approver and or the assignee can change the status and approve the output. So back in this grayed out field, we see all of the different materials that were requested. We've checked mark which ones we're going to approve or not approve. So then that will pop up this field here, and what we're interested in filling out is this drop down yes or no. So let's say that we approved these materials here. We had enough material, so they were aliquoted or what have you, and they're ready to go back to the requester. However, these last 2 samples or material, we did not have enough to fill the request, so we're going to reject that approval and say no, we do not have that to send back to the requester. From there, the approved task will be marked as completed, and then we will the unapproved task will be marked as canceled. Now that we have the new material, we need to give it a location in Benchling. So we have two different options. The first option is that we move it to an existing container. So we've added these create new container tables into this entry, and we will enter all the required information. So that includes the source container, the destination box, and the destination position. The second option is that we need to register a new container. So again, we have a similar table inserted into this entry, and then we will just enter the required information, which includes the source entity, the destination box, and the destination position. And then we will click transfer, and we'll finish the material request by completing the material request output. So here we can see that that table from the previous slide is all filled out. We created new containers, so we have the destination box, the position, and the destination container was autofilled because we were creating those new containers. So then down here we have the outputs for the material request. So we are just going to copy and paste the destination container to the container down here, and then click submit, and your material request is complete. That concludes our Benchling demo. Thank you so much for following along. If you're curious about any of the Benchling applications and would like to implement them into your own Benchling environment, please reach out to us at DigitalRadius.com. We'd be happy to support any of your Benchling projects. Thank you again, and take care.